Hello everyone, and welcome to this live stream. Today we'll be talking about organoids, and specifically organoids for animal health research. So really fascinating topic, really something new, because obviously organoids are used in, or at least we hear a lot about them. There's conferences and a lot of material, especially for humans, but they are quite fresh or new to the animal research field. And today we are here with a special guest. Um, let's see, switch to the next screen. We are here with Marina Canadas Ortega, that joins us as lead scientist. And Marina comes from the University of Salzburg, where she has a quite long experience dealing with organoids and researching on organoids. And we will have a little bit of an discussion today. So, Marina, thank you very much for being on the stream today, and welcome on this Friday afternoon for Europe and morning for the US.
Thank you for inviting me.
Welcome. Maybe let's start with a couple of basic information for, who is not quite familiar with what organoids are and what they're used for. So what are organoids and how do they differ from a traditional 2D cell culture or other 3D culture models, like if that for that matters?
So basically, organoids are miniature and very simplified versions of the organs that we're able to grow in the lab, and they grow from stem cells. So they are able to self-organize and replicate some of the structures of the real organs. That's the great thing about them. So how do they differ from other models? Well, from 2D, they differ basically because they are 3D instead of 2D. But from other 3D models, it depends. Other 3D models are not so able to replicate the tissue architecture. Like, for example, spheroids that are just accumulation of cells. And they lack the complexity of the organ structures and also the stemness. So organoids are more accurate in the functional representation, basically.
Okay. Maybe we will touch on this then later on. It will be interesting to understand, what are the differences between spheroids and organoids. You said something about this, but maybe you can explain first a little bit how they are created. How do you produce organoids in general?
So basically, they are created from stem cells. So you need to have a source of stem cells that they can be either embryonic or adult stem cells. And basically, you need to isolate the stem cells from the tissue. Usually, you get them from patients, usually. And then you need to culture to isolate these stem cells and to culture to maintain the pluripotency, and basically then you expose them to growth factors and you make them to differentiate, and there that is when they start to self-organize in 3D and to replicate this tissue structure. And to do that you usually use gel-like matrices, like matrix gel usually, to replicate kind of the extracellular matrix. And then they need to maturate for some time to be useful. So it depends, but they take usually several weeks to be able to differentiate and to maturate.
And is it enough to, take the stem cells, put them into matrigel with some growth factors and wait, or is there any other kind of technique needed?
Basically, I mean, once you isolate the cells, you need to be very careful to maintain the pluripotency. That's the main step. But then, they are very able to self-organize.
Okay. And how do you check for the pluripotency?
How do you check the pluripotency? Well, it depends on the type of stem cell, but basically you need to. It depends really. For example, I am more specialized in the gastric organoids, and you need to check that they are always are responding and weaned to keep the pluripotency. Because if you remove them, then fully differentiated, and they are not really useful.
And what do you mean when you say responding to wind? Like wind is a growth factors, I guess.
Yes.
So you
Yeah.
give wind, and then what is it that you're measuring?
So yeah, you can check several factors, several markers for pluripotency,
Ah,
and
okay.
then yeah.
And do you normally do this on like every single culture that you do? Like is it's a kind of a in-process control, or is this that you do once and then once you have established it for the particular protocol, then you just assume that it works?
You should do some checks from time to time, but in principle, once they are working, they work.
Okay, okay. And so let's come back to the other question. So what's the difference between an organoid and a spheroid?
Basically, spheroids are just accumulations of cells, and they don't have stem cells on them, while organoids do have the stem cells, and then they are able to differentiate and to form really so they mimic the tissue architecture much more than just a clump of cells. It's the main
Okay.
difference.
So are there like differences in the kind of applications that I have for spheroids, for organoids? Like if I take, for example, a tissue where I already have all the differentiated cells, the different types, and I just let them clump together and create a spheroid according to your definition. Is this, do I have the same kind of applications as an organoid, or is it something completely different?
It should be different because then it's less accurate. It's not keeping like the tissue architecture, so it's not exactly the same. So it's not so accurate.
Ah, okay, okay. I mean, this is an important point also, the structure itself.
Yeah,
Um,
and also in an asteroid you have many, so less different types of cells because they are already fully differentiated.
all right, all right. So it's a less types of cell that you can get, or a more limited, um,
Yeah.
let's say, yeah, how do you call it? Diversity of the different cell types that you
Yes,
can get
exactly.
out of that particular sample or that particular tissue, and then also the the organization is different.
Exactly.
And okay, but still, like, do you like if I'm thinking about other methods in 3D in 3D cell culture, where I don't know, you might want to now at the moment you see a lot of tissue engineering kind of methods or microphysiological systems or whatever, where the cells are embedded in a matrix maybe, or matrix or a scaffold, being this natural um, or synthetic. No, it's also used to give some kind of structure. What is the role? What is the role of a scaffold or this kind of structure? Do you have examples from your previous work on how this structure influences then the function also of the organoid?
Well, I think it really depends on the application. So, for example, if you are studying epithelia, you want to have like really epithelial structure. And
Mm-hmm.
for that, there are, for example, I have experience in skin grafts. So we used plasma from patients to create like a gel in which we embedded the fibroblasts, and then on top we would put the keratinocytes. So we were like really mimicking the skin. Um, in my previous lab, I was also contributing to working with a model of epithelia for the stomach for
Mm-hmm.
investigating infections. And in that, we developed like mucoses. We didn't develop them, but we implemented them in the lab and we used them to study the infections. And then you needed like a transwell to contain so to support the cells. And then they were able to grow in a really stratus epithelial, which was more significant for us to mimic the epithelium than, for example, a 3D organoid in that aspect.
Mm-hmm. Mm-hmm. Mm-hmm.
So it
Okay,
really
okay.
depends on the application on what you want to do.
Now, obviously, I, I guess that so organoids in general they used at different phases during the preclinical development of a drug. They obviously used it in basic research for a lot of different applications. But then, if we want to go a little bit more into the specifics, into the details of developing a drug. In this case, we are talking about developing a drugs for animals. Then we are a little bit more, let's say, bound to a more concrete kind of aim, and so we would have a more definite set of tests, that kind of phases also in our development. Sooner or later, for the development of a drug, we will have to go into animal studies, as we do for humans. In case of development of drugs for animals, we are, the animal studies are directly the clinical studies basically for the animals. And we've seen in the past that there are, I mean, the animals, the medicaments that are available to animals are not as abundant as the humans, and obviously because there's this missing step in between of a third, like, kind of model where to test all the toxicity and so on. Then we basically have the case that a lot of the drugs are tested on animals, but they're still quite toxic, or they have efficacy issues, or whatever. But what do you think are the kind of applications that would help improve also the safety towards animal trials in general? Or in which kind of cases can we use more of these models of organoids and less of the direct testing on animals?
So yeah, I think they can be used quite abundantly in the animal health research. First, they are basically they can up to certain point replace animal testing, because they can provide a platform in which you can test a variety of, I don't know, shampoos or these kind of cosmetics. They can for sure be gone with the use of epithelial organoids of humans or for animal or of animals. But then organoids have like a very wide range of uses in the clinical, in the preclinical research. So, for example, they can be used to model specific animal diseases, and also to investigate how is the zoonotic process, so how the different parasites or viruses or bacteria are able to colonize the animal, and then how they jump from the animal reservoir into the humans. So they can be very useful for researching infectious diseases, but they can also be used for drug testing to assess the efficacy and the safety of different types of drugs that can be tested instead of so you can either use the human ones for human and reduce some animal testing, and then you can of course use the animal ones for animals to make sure that they are safe and efficacy and efficient. Well, that they are
Efficacious.
safe. Efficacious, that's a word.
Okay. I mean, I can imagine that, especially the field of infectious diseases, it's one of the ones probably that would benefit the most, because it's quite dangerous, and I mean it's quite dangerous for the patient itself, and it's obviously also not the most easy to test. I also guess it's probably better or easier to control an infectious disease in a lab rather than in the final animal.
Yeah. No,
Um,
we have some examples already
okay.
about how you can use organoids to understand the infectious disease, either because it's dangerous or because it's like really mortal for the animal, so very fatal. So then you get you kill your model.
Mm.
So yeah, we have some examples in the feline infectious peritonitis disease. So it's
Okay.
caused by a coronavirus, like the virus, the most famous one lately,
Okay. Yeah.
and basically super deadly. So yeah, in order to understand the interactions with between the virus and the hosts, organoids were used to understand this replication, and they were in fact used for developing the main vaccine, or the only vaccine that is used right now, and for
Mhm.
the only little treatment that it has. It has a very complex number, like yes something something, I don't remember the the number, but it's the most prominent, or most prominent treatment that is the most prominent solution that is right now in the market. And another example I read about some nematode parasite in the calves,
Mhm.
in which they used organoids, the intestinal organoids, to understand how the nematode was colonizing the intestine, and
Mhm.
then they try to do some research and to develop a vaccine for that.
Okay. Okay. Really, really interesting. Really interesting. Give me maybe just an idea. So, if you were talking about the structure before, and in I guess that most of the people, if they think about an organoid, they will think about something spherical.
Hmm. Yeah, most of
How
them.
does it look like? Like what's the in, what's the out? How what is the kind of testing that I can do there?
So yeah, basically most of them are with the lumen inside. For example, that's very common in gastric organoids. That's something that difficults the research in gastric organoids, because the polarity is like in. So they are facing inwards, and then it's a bit complicated. So the inside part of the organoid will be the stomach.
Mhm.
So that's why it's a little bit hard to investigate this. That's why some groups have been doing research to invert the polarity so that the
Mm.
lumen is out. But the easiest solution that has been found right now, it's the develop of mucosoids, which is, as I described, like a transwell, and you have like the cells are in the base, and they are like forming the epithelial, and then they produce the mucus on the upper part. So
Mm.
that's very convenient to investigate this kind of, for example, the gastric or the intestinal diseases.
Mm. And so you need a particular kind of plate for this.
Yes. So transwells are like kind of tiny bases for them.
Okay, okay. So you use the normal cell culture inserts,
Yes,
like
exactly.
96 wells or
Mhm.
whatever.
Yeah.
Okay, okay, interesting. And you use this to recreate like the internal part
Yes.
of
Yeah.
the stomach.
In general, for example, brains are like round. Most of them are round. Yeah.
Okay, okay. And I guess if you have the lumen inside, then you would have to enter, I don't know, with a pipe. I know that they have these kind of things because they do
Yeah.
in vitro fertilization, but I don't know how practical they are.
It's not. It is not.
It just
That's
sounds
why
like
it's much more useful in the mucosoid form.
okay, okay. Um, which sounds obviously much easier to use and to handle. Did you see during the past years like also an evolution on the way organoids are cultured, developed, used in research in general?
Yeah. Um, I think that first we are advancing in the complexity of the models,
Hmm.
and the more with the advances in stem cell biology, the more we understand about how to keep the stemness of the cells, the more complex systems we can build. And also we have like a lot of advances in combining different types of cells, so they resemble more accurately the organs. And there have been also advances in integrating the endothelial cells. So trying to obtain somehow or to mimic somehow the blood vessels, so to perfuse them with to increase the nutrient and the oxygen delivery. Have been also quite some advances in the microfluidic field to allow the culture of these organoids in microfluidic devices, and they are called organ on chips.
Mhm.
And they allow a really tight control of the environment, so they are very useful to analyze different amounts of drugs, for example. So they also allow multiplicity or scaling up.
Mhm.
Um, what else? In this field, yeah, well, we had the great advance with patient-derived organoids to kind of analyze specifically for which patient or which drug would be more convenient for the different patients.
Mhm.
And all these organoids can be edited with CRISPR-Cas9. So that also has increased or has pushed this field forward.
I guess really important also in the case of rare diseases, where you may not have thousands of patients to to test
Yes.
your drugs in clinical studies. So you may want to have a kind of system to test the different medicaments beforehand.
Yes, exactly.
And what do you think that, like, we talked about a lot about the different, the potential of the technique, about what is the um, some of the different applications, what are different uses, the structure, and so on and so forth. What do you think are the limitations of the organoids? Is this a model that people should use? If yes, why? And what are the limitations on the other hand?
Mhm. So, of course, they provide a really good platform for a variety of things. So, drug testing or patient derived organoids to analyze the personalized medicine, all this. They have some limitations, of course. Some of them are more scientific, some of them are more practical. So, in the scientific point of view, they are still limited. They are not they don't mimic completely the functional complexity of the organs, and they are also very limited when you try to integrate or investigate more systemic approaches, because you can add some immune cells, or replicate a little bit the vasculature, but not fully. The immune system and the vasculature is not fully replicated, so it's limited in that aspect. So you
So
cannot
far.
model systemic interactions. And then you have problems that they are, in a more practical point of view, they are a bit variable between batches, and they require some expertise to be able to reproduce the results. And also they are a bit expensive, so that's a problem for
Okay.
some labs.
Like yeah, between growth factors and matrigel, I don't think that they are particularly cheap. Okay, this is also one of the things that that might be developed in the future. In the end, it's a commercial kind of question. But if some of these materials become commodities, maybe there will be also new suppliers of other alternative
Yeah.
materials. But the question is obviously also, how to make this, if this is really um, a valid, you know, kind of tool platform. This is maybe something that more people would like to know, and to use. What are kind of some of the issues or some of the things that people that never used organoids and would like to start using them should, you know, keep in
Be
consideration?
So I think that first they will need to understand if it's useful for them, because I mean they are like very modern and very famous now, but they are not the key to everything. So instead of going directly into just using them randomly, first you will need to understand if you really need them and if they really are good for your research. And then you need to basically understand basics. So how you do derive them, how do you culture them, the uses that they have. So you need to invest a bit in training and expertise. I will also recommend to like focus on the standardization and validation. So you start with small pilot studies and then you validate and optimize them, and you really need to focus on standardization. And yeah, if you are new, I will tell you, like from a personal point of view, to be patient because they are sensitive, they are a little bit picky. So you need to like keep going. At some point you will improve your technique. You will know if they like to be pipetted 3 times instead of 2, or 2 times instead of 4. And you will learn if you have the media outside for too long and it's not the perfect temperature, they won't like it, or things like that. So at some point you start troubleshooting, and you need to be careful of what you do, have that in mind, and adapt consequently. Like, this didn't work, try this, or check your media, check your growth factors that they are good quality. That's important.
So I hear from what you're saying that the learning curve is not particularly flat. It's quite steep in this case.
It is. So I was really lucky to have a mentor that really helped me understand how to work with them in the in the University of Salzburg, but also before I was doing skin grafts, which were also tricky. And people took the time to tell me, okay, what I know about them is that, for example, with keratinocytes for the skin grafts, you really needed to be like super careful in pipetting, or when shearing the gastric organoids, you also will need like to pipette 7 times with a certain amount of force, and it takes time. So there are great people out there. Follow them, ask them. Sometimes they provide courses. If I was really lucky, and Francesco Boccellato came to our lab to train us, not to and to train the people in the university. So if you are in a laboratory and you have the opportunity to host one of these courses, try, because it's very useful.
Yeah, yeah. Thank you very much for this for this kind of insight. What maybe some final words on the topic. What do you think are the the factors that are holding people back? What would we need to have a more widespread use of organoids also in animal health research? Or what could be the factors that are holding people back at the moment?
The limitations in from the scientific point of view, I would say that is something that holds some people back. And so you need to understand how good is they are for your research. And also the variability and cost are also holding some people back. Also, there are some ethical concerns because sometimes they are obtained from embryonic cells. So yeah, and you need really. I am not so sure how it is in the animal part, but in the human, if you have cells from human, you really have these ethical concerns and all these regulations that sometimes are not the easiest to navigate.
Yeah. I guess it's quite similar also for
Yeah, I will say
the
so.
animal field.
In fact, with the
Obviously.
animals, you have the additional part that animals cannot consent. So that's something that probably regulations need to put some work into regulate this.
Yeah, yeah, definitely. I mean, you have the consent of the owners in case of pets or whatever,
Yeah.
but it gets a little bit more complicated with other animals. Yeah, this is also why ethical committees are regulating all the animal studies,
Yeah.
and yeah, this is obviously a quite disputed topic. Okay, great. Thank you very much then for your insights, and thank you very much for being with us today. I would say we can move to a short Q&A. Let's see if we have some questions coming from the audience. If you have any question, you can obviously post it in the chat. We should be able to see them. We'll try to answer live. Let's give a couple of seconds to this. All right, I don't see anything. A lot to do with this Friday afternoon or morning. So I thank everyone for being on this live stream. Thank you very much, Marina, for being on the call. And
it was my pleasure.
Let's see each other in the next live stream. Bye-bye.